What I'd like to do is go over this naming convention again. Last time we had talked about the omega naming and the delta. I'm going to go over a few more examples of that. Okay, so most of you have heard of omega designation for fatty acids. That's how many carbons in from the end is the first double bond. It says nothing about how many double bonds there are. It just says the first one is so many carbons in. Nutritionists usually use that nomenclature. However, biochemists don't usually, unless they're conversing with nutritionists. They use this other designation, which is the delta. And that tells exactly which carbons the double bonds are on. And secondly, they count from the acid end, carbon number 1. So for oleic acid, the quote unquote healthy fat, it's going to be starting from the acid end 1, 2, 3, 4, 5, 6, 7, 8, 9. This says how many carbons. This says how many double bonds. And then where they're located is on the delta. Now, if there are no double bonds, for example, like palmitic acid here, it'll have no delta signature at all. It's an omega 0, 16 carbons long. Now, the other thing is there's a common name and then there's an IUPAC name. The IUPAC name is going to be a combination of two parts. They're going to have a deci in there if it's below 20. And then it's going to have another prefix in front of that to say how many above the 10. So in the case of oleic acid, it's an 18 carbon acid. So it's going to be octadecenoic. Okay. Because that's an alkene. And it's at position 9, which corresponds to the delta. Now, this list are the acids you need to know. The two most common ones on here are oleic acid, olive oil, linolenic acid, which is the most common omega-3, stearic acid, which is what you get from meat, and then these two down at the bottom, the DHA and the EPA. Those are both also common omega-3 fatty acids. Now, your test is going to be next week, test 3. And it'll be on Thursday. So Tuesday, I'm going to have a review. Thursday will be your test, in-class test. Okay, are there any questions about this naming convention? It's very different than what you've probably seen ever before, especially this delta. Okay, so let's jump over. Let's talk about lipids now. Lipids are really some of the older biological molecules studied, and that's why most of the lipids they go by common names because they were named before the IUPAC came into being. There are two main classes. One is saponifiable. That means it's got an ester in there, or non-saponifiable, meaning there's no esters. The non-saponifiable ones are like things like steroids, prostaglandins, those are some of the lesser known, particularly prostaglandins. Prostaglandins are produced from when you take an NSAID pain reliever like aspirin or ibuprofen or something like that. You're producing prostaglandins, and prostaglandins, there's quite a few of them. Some are bad for you, like for some of them prevent your blood from clotting. The other are anti-inflammatory. So there's good and bad. And right now, when you take one, it inhibits the production of prostaglandins, but it also inhibits the good prostaglandins. And there are two pathways to get there. And they're trying to develop drugs, over-the-counter drugs, that work on the bad one, but not the good one. So, okay, and the saponifiable ones, again, are the ones that have esters in it. There are simple ones and complex ones. When we were making soap, we were working with a saponifiable triglyceride. Okay, so the simple ones are basically a plain old acid and an alcohol. That's all. And the alcohol is usually a 3-hydroxyl group alcohol. And then the fatty acids are usually C18 length. And that can be any of the C18s. They can be plant-derived, animal-derived, it really doesn't matter. The complex lipids have other things in it, usually phosphorus compounds, and usually a nitrogen compound, besides the fatty acids and the alcohol backbone. Now, the thing about fatty acids, they're naturally an emulsifier. In other words, they naturally contain a very polar end and a very non-polar end. And that's what's required of an emulsifier. So if I ask you on a test, like I show you a molecule, is this an emulsifier? What you look for are two things. Is something on it polar and is something non-polar? If the answer to that is yes, then it can act as an emulsifier. Sometimes it's a really good emulsifier, and other times it's marginal. But if they have two of those parts, it can become an emulsifier, which is soap, detergent. They also call them surfactants. Another interesting phenomenon of fats, they form micelles. And what that is the parts that are alike grouped together, the parts that are not alike grouped together. So in a water environment, a polar environment, that's what the micelle will look like. In a non-polar environment, it's going to be exactly the opposite. Okay, so this is polar stuff out here. Hence the polar part, the surfactant, and the polar solvent are with each other. Now, if this was in like pentane, which is a very non-polar solvent, what you'd end up with is you have this thing turned inside out. So the nonpolar parts of the fatty acids, the blue part here, is going to be in the outside. And on the inside is going to be the polar part. Now, when you mention fatty acids, there are certain characteristics of fatty acids. Rarely are they ever branched. Normally, they're long, stringy things. There are different classes of fatty acids. There's short chain fatty acids, medium chain fatty acids, and just plain old fatty acids. So when you hear the word fatty acid, it's a long one. It's from 16 to 20. Medium chain ones are found in a lot of health food stores. And your bacteria and your microbiome in your gut produce short chain fatty acids when they work on their prebiotic food. And it's always even. And the reason why it's always even is because we synthesize fatty acids by 2 carbons at a time. That's why they're always even numbers on the fatty acids. And sometimes there's no double bonds. Sometimes there are multiple double bonds. Sometimes there's like 6 and 7 double bonds. Just depends on the fatty acid. Now, stearic acid is a long fatty acid, 18. You get that usually from meat. Oleic acid from olives, linoleic acid from corn. When you have a double bond in there, the natural version is called a cis. It's not a trans, it's a cis. Cis means they're on the same side. Carbons are on the same side as opposed to being across from one another. When they're on the same side, it ends up kinking, putting a kink in the long chain. So if you have multiple kinks, it's going to make it look like a fishhook. Usually in biological books, they'll show a membrane as having straight chain like this. And that's only with stearic acid, a unsaturated stearic acid does not look straight. And none of the textbooks I've seen have them as a kink in them. Now, it's really important on your nutrition to not eat trans fats. And the reason for that is because our enzymes are looking for kinks in the backbone and the carbon backbone. If there are no kinks, the enzyme does not work as well. So if you used to be Crisco shortening was designed to eliminate using lard for baking. But when they made Crisco a long time ago, they produced a ton of trans fats. So I was raised on trans fat. My mother baked all the time and she only used Crisco. She rarely, if ever, used lard. And I'm standing here alive. So it's not going to kill you, but it's not good for you either. Okay, when you start putting double bonds into the long fatty or the long carbon chain, the physical characteristics change along with things like melting point, boiling points, the texture. Long straight chain fatty acids tend to slide easily against one another. So they tend to have a better mouthfeel than ones that are highly crooked, like polyunsaturated fats. Now, there's a term called essential fatty acids, essential amino acids. Both those are meaning that you cannot Our body doesn't make them. We have to eat them somehow. So if you're a vegetarian and you're eating a lot of vegetarian protein, just make sure you have a balanced amino acid set in there, because plant amino acids are different than animal amino acids. So they produce different ones. So you usually have to blend them together to get a balanced set. Now, omega-6 and omega-9 fatty acids are easy to come by. It's the omega-3s that are hard to come by. Two sources for those: flaxseed and fish that swim a lot, not bottom feeding fish. So things like salmon, mackerel, tuna, sardines, anchovies, all those fish are omega-3 fishes. Now, you've heard of fats, you've heard of oils. Fats are usually solid at room temperature. Oils are liquid at room temperature. Fats are usually saturated. Oils are usually unsaturated, just in general. Now, some are kind of on the edge, like for example, if you go back up here, you can see how the temperatures differ quite a bit in terms of their melting points. Okay, so this is the most common type of fat, a triglyceride. Tri meaning three. We have three fatty acids, and this is the alcohol over here, 3 hydroxyl groups. This is called glycerol. And what binds the acid to the alcohol is an ester. So we have 3 esters there. Now, an ester can come apart in an acidic environment or alkaline environment. If you introduce an alkaline to a triglyceride like we did in lab when we made our soap, what happens is the ester bond is cleaved and you end up with regenerating the alcohol and regenerating the acids. And because it's in an alkaline environment, we end up making the acid salt. So this ends up being extremely polar. Because it's ionic, you cannot get any more polar than that. So that's why when you make soap, you want to use a nice long chain to get that non-polar tail there. And then you're going to use a very harsh alkaline environment to cleave that ester. And what you end up with is an emulsifier that's got a very polar end, ionic, very non-polar, straight chain. So it works really well as an emulsifier. Now, the way you make diesel, biodiesel, is you take fat and you do something called transesterification. What that means is you replace this with methanol, a very simple alcohol. You still have the long tail there. And that's why when I was growing up in the 60s, you'd see these, particularly along the coast, these pink buses with big flamingos painted on the side, and it says powered by biodiesel. And you can make it in your garage. It's very easy to make. And used to be, all the restaurants had a bin out in the back where they put the old grease. And they would pay someone to come and take it away. And that's what they would use to make biodiesel, is that cast off grease from restaurants. And it's very easy to do. That would be a good lab for us to do one of these days. Okay, so whenever you make an ester, you generate the ester itself plus water. So these water molecules come off whenever you make an ester. Now, the enzyme that digests fat is lipase. And when we get to our enzyme chapter, we're going to go through a number of enzymes and how to name them and stuff. But lipase, and it's located in your small intestine, and it requires generally a neutral pH, like physiological pH is 7.4, so it's around that neutral area. It won't work on your stomach. It's a very acidic environment, pH of 2. It works fine, though, in your small intestine. Now, when you say soap, it's generally assumed that it comes from a triglyceride as opposed to a detergent, which does not come from a triglyceride. They're both emulsifiers. They both have a very polar end and a very non-polar end. They're just not the same. So whenever you say soap, it's generated from a triglyceride. Okay, so the base you use to saponify things, it will vary depending on what kind of base you use, what you end up with, whether it's liquid or solid. So we use sodium hydroxide in our soap lab. So that's why it produced a hard cake soap. If we use potassium hydroxide, we would have got a liquid soap, similar to dishwashing soap. Crisco used this process to turn polyunsaturated fats to saturated fats to mimic lard. Lard is a very non-saturated fat. What happened, though, you frequently created trans versions. It converted all the cis to trans, and that ended up with a very similar product to lard. So if you have an ester in the presence of hydrogen, it will convert will hydrogenate the double bonds to single bonds. Now, nothing happens if you're starting with a saturated fat. Nothing will happen because it's already hydrogenated. In its acidic environment, you end up with the protonated fat, protonated acid. In an alkaline environment, you end up with the acid salt. Okay. Now, all of you have heard of a wax. This is what a wax looks like. Basically, it still starts with a fatty acid, only the alcohol is very, very, very long. We're talking, like this one up here is 16 carbons long. It can be up to 24 carbons long. But it's basically really long alcohol and a really long fatty acid come together via an ester bond. And because of this, they tend to be very hard at room temperature. Now, a phospholipid looks very similar to a triglyceride, only instead of having 3 fatty acids, one of the ester bonds is with phosphoric acid and an amino alcohol, like choline or something like that. So this is an ester, and that's an ester. Phosphoric acid has 3 it's a triprotic acid, so it has 3 hydrogens that can come off. So each one of those is capable of doing an ester bond. This is also what ties together DNA bases, is also this phosphoric acid. It's very common in our bodies. Frequently, cellular membranes are made of phospholipids, and this is what they look like. Okay, so 3 carbon backbone, that's the glycerol, 2 fatty acids. The phosphoric acid here has got a double ester. That's what glues this amino alcohol to the glycerol backbone. Who's heard of lecithin? Anyone? Lecithin is found in egg yolks, and it's a phospholipid. That's why you use egg yolks to make mayonnaise, because you need an emulsifier when you make mayonnaise, and that comes from egg yolks. So choline is very, very common in biological systems, and it's got an alcohol on one end and an amine group on the other end. The alcohol is the one that makes the ester. And this is what lecithin looks like. So this is the choline down here. And these are any 2 fatty acids. Now, there are a number of structures you need to know for this test. Choline is one of them. So you need to be able to put together lecithin. This is another phospholipid, slightly different. What's different about it is not using choline. It's using an amino acid. And these are some other kinds of complex lipids. And these are used a lot in the neurological part of mammals. Basically, it's just a different backbone. So if you look at this particular one, notice we have choline down here, and we have phosphoric acid, And this is the backbone. Now this bond here, that's unit 4. That is similar to an ester, only it uses nitrogen instead of oxygen. It's called an amide bond, A-M-I-D-E. Sometimes there's a carbohydrate bonded in there as well. And if that's the anomeric carbon, is that a reducing sugar? Remember what determines a reducing sugar? Whether you have a hemiacetal present or not? Do you see a hemiacetal there on the anomeric carbon? No, it's a full-blown acetal, so this is not a reducing sugar. Now, steroids are also lipids, but they don't have an ester bond in it, so they're not saponifiable. This basic system of a steroid, you need to know this. Okay, it's got 3 6-carbon structures and a 5-carbon structure. So if we look at, so here's 1, 2, 3, 6-carbon rings, 5-carbon ring. And then there's various stuff hanging off of it. We have a whole bunch of steroids in our body. And they're used frequently in medication for your body. Now, you've all heard of gallbladders. Bile is stored in your gallbladder. This is what bile looks like. It's also a steroid. And it acts as an emulsifier. Why? Because it's got a very polar end over here and a very non-polar end over here. So it acts, that's why bile is used to help digest fats in your small intestine. So this works in conjunction with lipase to digest fats. So what happens is it breaks up fats into really, really small pieces, increase surface area so the enzyme can work more effectively. So this is arachidonic acid. It's 20 carbons. And it's a precursor to prostaglandins. And these are the ones that good and bad. And this there's an enzyme called cyclooxidase 1 and cyclooxidase 2 that works on your arachidonic acid and forms the prostaglandins. Good ones and bad ones. They're very similar. Those are the 2 differences there. Makes all the difference in the world. Okay, so these are fatty acids. And these are the lipids. Now, in steroids, all you need to do is the basic 4-carbon cyclic rings. And an example of a phosphoglyceride would be lecithin. Okay, so the hard part on lipids are the structures. Unit 2 structures were challenging as well, but the fatty acids are the tricky ones here. And putting those together with into 3-carbon backbones, et cetera. Okay, so that's what I wanted to cover today, are lipids. And my graduate work was done with lipids. What I did is they were in the back in the 60s, they were labeling, starting to label fats as cis and trans. And that's what funded my research, was that legislation, actually. And I used a machine called a gas chromatograph, actually tell the difference on how many cis and trans fatty acids were in a particular product. So, okay, let's see. Let's start lab early today.